And I will talk in particular about non-coding RNA, sorry. Something that concerns long non-coding RNA that everybody thinks is useless. It comes from portion of the chromosome that are not being sequenced. They tell you that the whole human chromosome has been sequenced. It's not true. Millions and millions of bases coming from repeated regions of the genome has not been sequenced because the machines are unable to sequence these regions. But I will tell you a story that concerns the stress-induced modification that happens in only human cells nuclei when they undergo some stress treatment. And by stress treatment, I mean heat, shock, heavy metal treatment, osmotic shock, UV, and whatever you can imagine that can damage the cell metabolism. And you will see that heat shock causes the transcriptional activation of constitutive heterochromatin, which is normally silent, transcription inactive. This cartoon represents the fate of coding RNA. And the reason why I show it, because many of the proteins that will be, that you will see in activity in what happens to non-coding RNA are the same proteins that are bound to the coding RNA. So this cartoon is very important because it shows what happens to the RNA coding, messenger RNA, pre-messenger RNA. This transcript is covered by a number of pre-mRNA HNRNP proteins, heterogeneous nuclear RNA binding proteins, and other family of proteins that are called SR proteins. There are RNA binding proteins that are called SR because they are rich in serine arginine in their carboxy terminal end. And so the RNA becomes a ribonucleoprotein fiber on which all kinds of reactions occur, particularly the splicing reaction that has been alluded to by John Abelson before, until you get to the mature form of the RNA that still is covered by proteins, the same protein as before, not all of them, but some of them. And please notice this EYC. So the place where there has been a splicing event, where the two exons join, right there is a protein complex that remains bound to it. It remains bound to it, and it is exported from the nucleus to the cytoplasm, where the RNA is translated, and some of this protein shuttle back to the nucleus after they've done their job. Now, I showed this cartoon, and please keep in mind, because it will come back often. The two families of proteins that will come back in many talks are this one. The so-called heterogeneous nuclear RNA binding proteins and the so-called SR proteins. The heterogeneous nuclear RNA binding proteins, this is a two-dimensional separation. You see there are 25, 30 proteins. The most best characterized is this one, A1, HRNP-A1. And I'm proud to say that most of the characterization of this protein has been done in our laboratory in Pavia when I was still active. Now I'm sort of retired. And then there are other proteins which are very important. And please notice this tiny protein here. These are small, basic. These are big, acidic proteins. This HAP was called HAP because it interacts with, it's a protein that it interacts with a 1 in the 2 hybrid system. But anyway, keep in mind these 2 proteins, A1 and HAP. The characteristic of this protein and also of the SR proteins is to have a modular structure. They are composed of different RNA binding domains followed by a domain, carboxyl terminal domain, which is different in different proteins, but the message is that the RNA binding domain or the RNA recognition domain binds to RNA, mediates the binding to RNA, while this other domain, which is in the case of A1 is glycine rich, mediates the protein-protein interaction. And the same is true for the SR proteins, which have been much more studied in the context of splicing, particularly this one, SF2-ASF. You see, it has 2 RNA recognition domain plus this domain which is rich in serine arginine. And all these factors, they all have this modular structure. Now, we were searching for a function of that tiny little protein HAP. Okay? We knew it bound to A1 in the 2 hybrid assay, but we didn't know anything else. So in order to find a function of a protein in a process, what do? You perturb the process. And we knew that heat shock, for example, the thermal shock, so you submit cells to a thermal shock, 1 hour at 42 degrees, or 5 minutes at 45 degrees, and then you bring back at 37 and see what happens. And it is known for a long time that, for example, heat shock, which is the best characterized shock treatment, heat shock shuts off completely, almost completely, DNA replication, transcription, and splicing. It shuts off transcription almost completely, except for it induces the transcription of the so-called heat shock proteins, genes. So what happens is that in heat shock, all the transcription is shut down, except the transcription of few proteins, which are the heat shock factors, that is induced by heat shock factor 1, heat shock factor 2, and so on. And they produce proteins that are necessary to protect the cellular proteins from misfolding due to the heat. So they are chaperone proteins. But apart from that, everything is shut down. And also the splicing is very severely affected. And so we decided to submit to this treatment to the HeLa cells or other types of cells, and we discovered something very funny. That when we did that, and we looked at the by immunofluorescence with antibody against this mysterious protein, okay, we discovered that this protein, after 1 hour at 42, followed by a recovery time of 3 hours at 37, rather than being distributed uniformly in the nucleus, is concentrated in few bodies. No, huge. These are huge. They are something between 1 micrometer and 3 micrometer. And okay, as you know, in the nucleus, there are a lot of structures called bodies, okay? So the nucleus is not an amorphous gel. It contains a lot of structures called body, chiral body, coil body, speckles, all kinds of things. And most of these structures actually are related to the RNA metabolism. So they are stored, splicing factors, RNA proteins, they go back and forth, in and out. So most of these bodies are, as I say, related to the RNA metabolism, splicing, transport, and so on. These bodies, after many extensive characterization that I will describe, have been included among the bona fide nuclear stress body. And you see this on Cell by Spector. You see here our bodies are included among, as I said, the real stress bodies. So the properties of these bodies, which have been initially identified by using this antibody against this strange protein, are the following. So they peak after the heat shock, after the shock has ended and cell have been brought back to 37. It's reversible, so after several hours they disappear. It does not require protein synthesis. It does not occur at 4 degrees, so it requires energy. It requires RNA synthesis, and so on. They are novel nuclear structure in the sense they do not coincide, for example, with speckles. Speckles are concentrated as the SC35 protein, which is an SR factor, is concentrated in speckles, which are one of the bodies. And this is cells are colored with antibodies against either HAP, this protein, or against SC35, or against coilin. And the fact that you have red and green signals and no yellow signals, it means that there is no overlapping. So they are not I'm a bit confused. Anyway, so they are novel, and it says that they are not speckles, they are not coil body, they are something else. What are? We looked at the electron microscope. What the hell are these huge structures? And it turns out that they are clusters of perichromatin granules. Perichromatin granules are structures, they are tightly packed RNA molecules, which are stay near the heterochromatin, okay? And they form this enormous structure. You can see here at various stages. And what is interesting is that these new structures co-localize with heat shock factor 1. It was already known that heat shock factor 1, if you label the cells with antibodies against heat shock factor 1, you saw bodies. Okay? It turns out that our stress bodies are the same as those seen with heat shock factor 1. Heat shock factor 1 is the factor that I described before. You see, you have red and green color, and the fact that you have yellow color means that the two overlap. Now, the number of these bodies, before moving to that, another interesting thing we have noticed is that a subset of the splicing factor of the SR family is recruited to these bodies. Not all of them. And strangely, not HNRMPA1. HNRMPA1 remains diffused in the nucleus. But, for example, SRP30, and most important, this, SF2, and 9G8, this is another SR protein. So these three co-localize in these bodies, but for example, SC35, as I showed you before, does not. So only a subset. In fact, in these bodies, you can find among the transcription factor, HSF1, HSF2, RNA polymerase 2, and among RNA binding proteins, HNRMP-up, HNRMP-M, many SR factors, and this protein, SAM68, which you will be here in the next few days. So it's a selective recruitment of protein. The fact that in these bodies you recruit SF2 but not A1, which is very abundant, suggests that the formation of these bodies could affect alternative splicing. We know what alternative splicing is. Alternative splicing is something that happens when you have suboptimal splicing site, either accept donor or acceptor site, and you can choose between one or another depending on the equilibrium of the various factors involved. Okay? You will hear a lot about alternative splicing. But anyway, it has been known for a long time that, for example, A1 and SF2 have antagonistic effect on, for example, the choice between two 5' splice sites. This was an old experiment by, I think, Adrian Kreiner in Cold Spring Harbor. He observed that depending on the relative ratio of A1 to SF2, you choose the proximal 5' splice site or the distal one. Of course, if heat shock causes the sequestration of one, for example, of SF2, while leaving A1 undisturbed, you are bound to have a switch in the choice between, okay? And this is what happens. And this is what happens, and we verified, for example, by looking at this is a classical alternative splicing situation. It's a E1A antigen of adenovirus. You can clone it into a plasmid, and this gives several isoforms by alternative splicing that you can identify and measure. And if you look at the relative ratio between the different isoform of this protein, it's an adenovirus protein, you see that it changes during heat shock and during the recovery. And I think it's better illustrated. Okay, no. There was another slide, but this gives you the idea. So the formation of these bodies is bound to perturb the equilibrium between factors, splicing factors, RNA binding proteins, and is bound to change something in the... Now, I have to make a long story short. We observed that the number of these bodies changed. There were 2 bodies, if you look at the fibroblasts, for example, primary cell lines. But in HeLa cells, the bodies could be 4, 6. So it has something to do with the ploidy, indicating that the bodies were formed in correspondence to some chromosomal locus. The number of bodies was proportional to the number of chromosomes. You know that HeLa cells have a lot of chromosomes in addition to the normal number. And so we decided to find out which chromosomes, where on the chromosomes, were the seeds of these bodies. And so we made hybrids. We took hamster cells that do not form, and the reason why you will understand, and constructed cell lines in which the hamster cell contains one chromosome, the other chromosome, and so on. Only one human chromosome to identify the chromosomes that give rise to this phenomenon. And we found 3, chromosome 15, and chromosome 14, no, 15, chromosome 12 is not represented here, but most interesting, chromosome 9. Chromosome 9 is the most efficient. And the reason why we studied chromosome 9 is because in our lab we have a mini chromosome, this one, that happily lives in the cells with the other chromosomes, that is consists of the centromere of chromosome 9 plus the bands around the centromere. And these mini chromosomes, as you can see, in the human hamster cell hybrid, makes so we narrow down the locus where the responsible for the formation of the body to this region, to the centromere and the pericentromeric region of this chromosome. And in fact, this is an experiment that shows that this is the mini chromosome, and this is a probe that labels the heterochromatin, pericentrometic heterochromatin of these chromosomes. And you see that the 2 stress bodies are overlapped to the probes. While if you look at chromosome X, these are probes against not satellite 3, but against alpha satellite, another repeated sequence, and there is no overlapping between the bodies. So what happens? What happens is that this shock induces the production of polyadenylated satellite 3 RNA. So you have this enormous length of repeated sequences called satellite tree. This is one of the satellites in the cells. There are satellite 1, 2, alpha satellite, but satellite tree is one of the most abundant. And is concentrated in the pericentromeric area of chromosome 9. So, when you heat shock cells, something very funny happens. This region that is normally transcriptionally silent, and especially during the recovery after heat shock, there is an enormous increase of transcription of polyadenylated DNA that ranges from 10,000 base pairs to 1,000, very heterogeneous, but an enormous increase, which is surprising, because this satellite region is not supposed to be transcribed. And yet, the effect of heat shock is the transcription. But not only the transcription, an increase of something 40,000 fold of the normal, because this satellite region in fact are transcribed at a very low level. Nobody knows why, but there is a limited amount of transcription. But after its shock, there is an increase of 40,000 times. So we thought, well, that means that this region is no longer heterochromatin. And in fact, heterochromatin, constitutive, which is not transcribed, is characterized by being rich in repetitive elements, and that is okay. It's transcriptionally silent, doesn't contain acetylated histones. It contains methylated histones, is stained by DAPI, which is a stained nucleic acid, is associated with heterochromatin protein 1. So there's features which are well known, while euchromatin, that is the open form of chromatin, is being transcribed, is characterized by acetylated histones. So to make a long story short, we observe that in fact this region, the pericentrometic region of chromosome 9, constituted by satellite tree repeats, has all the features of euchromatin, active chromatin, in the sense that they are not associated with histone 3 methylated on K9, lysine 9, which is a marker of heterochromatin. They contain acetylated histones. They should not contain acetylated histones if it were euchromatin. And more important, they are associated with histone acetylated on lysine 8 and lysine 16, which are the tails of the histone acetylated on these two lysines are diagnostic of transcription, of open chromatin, transcribed chromatin. And this double staining, usual thing, confocal microscopy, shows that. So, you have this enormous induction of satellite tree RNA after its shock, which is asymmetric. So you have 30,000, 40,000 fold increase on one strand and only 10-fold increase on the other strand. is asymmetric, but both strands are transcribed, and this is important. Of course, having this enormous amount of polyadenylated RNA available after its shock, we could clone the RNA by using the usual trick. And we discovered what we already knew, that the satellite tree RNA consists of runs of these pentanucleotides, runs of 100,000 pentanucleotides, degenerated, not always perfect, terminated by a decanucleotide. This is a termination signature. But it's a mess. So a lot of RNA, repeated, divergent. But in any case, this fact is very important because this is rather conserved. It's important to make probes to study. Okay, this is another slide that shows that this RNA, using the oligos designed on the basis of the sequence we deduced, it in fact colocalizes, you see the yellow color, colocalizes with the stress body. So what is the model of all this, of what we observe? The model is the following. So you have satellite tree RNA, which is organized in heterochromatin around the centromere, millions of bases, and heterochromatin is indicated by a red color. Okay? When you heat shock factor 1, which sits in the cytoplasm normally, when you heat shock, it trimerizes, moves to the nucleus, and binds to RNA because the satellite tree RNA repeat, for some reason, has a recognition sequence for heat shock factor 1. A lot of recognition sequences because it's repeated. And so heat shock factor 1 binds, it recruits histone acetylase, for example, and it converts heterochromatin into euchromatin. At this point, RNA polymerase comes in as access and starts transcribing. You have RNA polymerase 2, you produce the RNA, and then binding proteins bind to this RNA, but this RNA is not properly processed. Okay, so it accumulates there, is not spliced, or is spliced but in an efficient way. In the end, it's round up in these granules, and these granules constitute, they get together and they make this cluster of granules, and they give origin to the body. The body stays there, doesn't leave the site of synthesis of satellite tree. After several hours, the body disappears, the RNA is degraded, and the region reacquires its heterochromatic features. Okay? This part is mysterious, how it happens, but I will talk about it a little bit. Okay, I will go very fast. You can induce this same phenomenon in different ways, I said. Not only by shock, but also by treating the cells with heavy metals, cadmium sulfate. Or or you can treat them with a hyperosmotic shock. So you incubate the cells in a medium full of sorbitol, a sugar. Cells get mad and they form stress bodies. But the hyperosmotic shock is sort of interesting because the same thing happens as with heat shock, but what happens is mediated not by heat shock factor 1, but by another transcription factor, which is called tonicity element binding protein. So this heat shock factor has a binding site in the satellite 3 repeat, like HSF1. Binds there and does the same thing. So it does not depend on heat shock factor 1, but on this other protein. Now, and this shows the colocalization of small stress bodies and tonicity element during osmotic stress. Nothing. Anyway, the level of satellite 3 decreases progressively during recovery from stress. But it lasts a long time. You see, there's hours, 2 hours, 6 hours, 12 hours, and so on. And we said, well, this looks like the ideal situation in which the degradation of this RNA is used to mediate the reconstitution of heterochromatin. Like it happens, it has been demonstrated, in Saccharomyces pombe. Here too you have the activation of centromeric regions or telomeric regions. You have symmetric transcription, and then the double-strand RNA that is produced is processed by dicer, and you produced small heterogeneous RNA which are recruited by this complex. So this is the RNA interference pathway, okay, which is yeast is used to because these fragments are complementary to the sequence where they come from, and they bring, for example, this protein SV16, and they reform the heterochromatin. This is demonstrated brilliantly. And we thought, well, you're pretty sure the same thing happens here with our RNA. And so we looked for the presence of short RNAs. What you should look for is RNAs of 20 to 25 nucleotides long, because those are those recognized by this complex. Unfortunately, we haven't been able to find this diagnostic small RNA. Instead, we find a ladder of RNA that goes from 100, 200, 150, and then down in the small RNA where we were hoping to find the magic 20 RNA. In fact, in our method, we can clearly see the microRNA 16, which is nothing. We have this ladder, which is very interesting because the intervals are 5 nucleotides, or like the repeat. So we don't know. We don't know, and right now we have no personnel and no money to pursue this research. Okay, in conclusion, you could think, after my talk, that stress is harmless. Each of factor 1 takes care of the integrity of the folding of the protein. The stress bodies reprogram the splicing by sequestering certain factors and so on, in order to activate or deactivate certain function. So the idea is that the modification of the alternative splicing is has the function to protect the cells during this critical time by reprogramming the splicing program. Okay, so that's fine. Everything is taken care of, the cells can go back to live happily, but that's not the whole story. In fact, stress, as you can imagine, is harmful. And I give you a glimpse only. For example, we have observed that heat shock affects cell progression and leads to accumulation of cells and G2M. It induces defect in the migration of the mini chromosomes that, in other words, the presence of this body that stays there, it never leaves is probably very harmful in cytokinesis. Why hamster cells and mouse cells do not form heat bodies? The reason is that they have satellite tree sequences, but they are not concentrated in a huge region like the one of chromosome 9, for example. They are spread all over the chromosomes. So probably there is activation of transcription in these regions, but you don't see them at the microscope. You don't see bodies. Okay. So this work was initiated 25 years ago when I was still active, and has been continued by my collaborators. And my only job is to talk about these things here and there. Thank you.
Okay. Thank you very much. Yeah.